All right, so we were talking about where the numbers come from in the phenotypic ratio. So down here, the three individuals that will be tall plants with yellow pods, that 3 represents the first trait is dominant and the second trait is recessive. And this is where the 3 individuals are. Big T, little g would give you a tall plant with a yellow pod. And here you have big T, which is going to give you a tall plant, 2 little g's, yellow pod. And then this last one, big T, 2 little g's. So you can also get 3 out of 16 if you do the first trait recessive and the second trait dominant. So short plant with a green pod, and those are going to be right here. Little t, big G, little t, big G, and little t, big G, little g. And the last individual in the Punnett square is going to be represented by 2 little t's, 2 little g's. So that means 1 out of 16 of the offspring are expected to be short and yellow, both traits recessive. Now, if you remember this ratio, 9 to 3 to 3 to 1, then if you know that what you're looking at is, let's see, oops. If you know that what you're looking at is a dihybrid cross and you remember this ratio, 9 to 3 to 3 to 1, that tells you that out of 16 offspring, 9 are expected to have both dominant traits. 3 are expected to have the first trait dominant, the second one recessive. 3 are expected to have the first trait recessive and the second trait dominant. And 1 out of 16 are expected to be both recessive. If you can remember that and you're given a problem to solve, a Punnett square problem to solve with a dihybrid cross where you're crossing 2 individuals that are heterozygous for 2 traits, like the big T, big G, little g, and it can be any traits. You've got both traits heterozygous. Then if both parents are heterozygous for 2 traits, this will always be the ratio of the phenotypes. Okay, that's the important thing. Now, so this is the experiment on the garden pea plants that Gregor Mendel used to determine the law of independent assortment. So he said it basically works like this. Let's say that you've got in the parent cell, you have 2 pairs of homologous chromosomes. And so here this the red would represent that it came from the mother, and the blue would represent that it came from the father or the sperm. Red is from the mother or the egg, and the blue is from the father or the sperm. So we've got this pair of chromosomes that contain the gene A. The A gene, for whatever trait that it represents, we've got a big A and a little a. So this individual's heterozygous for the A gene. And it's got a big A allele and a little a allele. And then the individual is also heterozygous for the B gene, and they have a big B and a little b allele. So what's going to happen is, and this is after replication, but you still have big A, big B, little b. You're going to end up with all possible combinations in the sperm or the egg that form. So you can either get big A, big B, little b, big A, little b, or little a, big B. This is just an illustration to represent the same thing that we showed right here, that we showed that you can get big T, big G, big T, little t, big G, little t, little g. When you have an individual that's heterozygous for 2 traits, and this is the law of independent assortment, it's saying that whatever trait A represents, that those alleles, the A alleles, are going to segregate independently of the B alleles. So you don't just get these two versions, big A, big B, little b, you also get a combination of big A, little b, and little a, big B. That's what the law of independent assortment states. Now, there's two slides on probability, and I will explain them to you, but I prefer Punnett squares. In the laws of probability, if we want to know the chance of, let's say that there's a gene that's represented by the letter A, and we want to know the chance of an individual passing on a big A. So the individual has big A, little a, right? So the chance of the individual passing, if this is the genotype of the individual, the chance of that individual giving a big A to their child is half, or 50%, because it's 1 out of 2. And little a is also half, or 50%. Okay? So the offspring, the chance the offspring will inherit 2 big A's is going to be both of those chances multiplied together. The chance of a big A is a half. The chance of another big A is a half. When you multiply them, you get a fourth. So the chance of the individual from a cross between 2 big A, little a parents, the chance of that offspring inheriting 2 big A's is 1/4. The chance of that offspring inheriting a big A and a little a in that order is going to be 1/2 times 1/2 is 1/4. And then the chance of the offspring inheriting a little a is still a half, big A still a half. So little a, big A is also a fourth, which gives you the chance of being heterozygous as 2 out of 4, or 1/2. And then little a is 1/4. This is the product rule, I think. I don't know if it's written up here anywhere, but it's called the product rule. And the sum rule allows us to add the genotypes that produce the identical phenotype to find out the chance of a particular phenotype. So we know that big A, little a, and little a, big A, and big A all are going to give us the same dominant phenotype. So if we add them all up, the sum rule, we get 3 out of 4. So it's a 3 out of 4 chance that we're going to get offspring with the dominant trait, and then a 1 out of 4 chance that we're going to get offspring with the recessive trait. This is a little much for some people. It's a whole lot easier to just take the 2 parents who are big A, little a, put their alleles on the outside of the eggs, can go on the outside of the columns, big A, little a, and then the alleles for the sperm can go on the outside of the rows, big A, little a, and do a Punnett square. And you can see the combinations that they form. 2 big A's, a big A, little a, another big A, little a, and a little a, little a. And that shows you that 3 out of 4 are going to have the dominant trait, and then 1/4 is going to have the recessive trait. This is one for albinism, by the way. So if you have 1 big A, you're going to have normal skin pigment, and if you have 2 little A's, you will not produce pigment in your skin, and you'll be an albino. Okay, now let's talk about test crosses. So a test cross is performed on individuals who have a dominant phenotype, and that is because we want to know if they are dominant, for example, big A, so that means there would be true breeding for that trait, or if they're heterozygous, which means that they carry the allele for the recessive trait. So we don't know which one they are just by looking at them. We know that they have the dominant phenotype, but we don't know what their genotype is. So what the test cross does is it crosses the individual who is dominant with a recessive individual. And based on the offspring that are produced, we try and determine what the phenotype or what the genotype was of the dominant individual. So test cross determines the genotype of an individual having the dominant phenotype. So here's an example of a test cross. We're crossing a plant that is tall, and this tells us that if the plant is tall and is a carrier, if the plant is tall and is heterozygous, carries the little t allele, then some of the offspring are going to be short. So we cross the dominant individual with a recessive individual, a tall individual with a recessive. And if any of the offspring are short, then those two little t's didn't just come from the short parent. One of those little t's came from the tall parent, if that makes sense. So if we get any offspring that have the recessive phenotype, we know the dominant parent is heterozygous, that they contain the dominant allele. We know that because they're tall, because they have the dominant trait. But they also are carriers of the recessive allele because they had some offspring that are recessive, and they had to have given one of those little t's. Okay, now if we cross the dominant individual with a recessive and we get all of the offspring have the dominant trait, more than likely that dominant individual is homozygous big T, big T. But it's not always 100% because these Punnett squares are just probability. They're just telling us what is most likely to happen. You know, you can have this parent that's heterozygous could be crossed with a recessive short plant and all of their offspring be tall just by chance. Just because these are the possible combinations of alleles doesn't mean that's exactly what's going to happen. But with garden peas, since they're producing 1,000 offspring at a time, you can be pretty sure if none of the offspring are short, that the original parent is true breeding for that tall trait. So that's and then this just tells you about a 2-trait test cross. I'm going to skip that. All right, let's talk about something besides garden peas. So section 11.3 is Mendelian patterns of inheritance as it relates to human disease. Genetic disorders are medical conditions caused by alleles inherited from parents. Now, when we talk about genetic disorders, we have to think about our 23 pairs of chromosomes. 22 of those chromosome pairs are called autosomes. These are chromosomes that are not sex chromosomes. So if there is a genetic disorder where the gene for that disorder is located on one of the first 22 pairs, we call it an autosomal disorder. Okay? So some genetic disorders are autosomal dominant, and that means that the individual who has one dominant allele has the disorder, but if the individual is recessive, like 2 little a's, they won't have it. So just one dominant allele will give you the disorder. They're not as common, but autosomal recessive disorders are much more common. In this case, only individuals with 2 little a's will have the disorder. In the autosomal dominant, if they're 2 little a's, they do not have the disorder. But if it's autosomal recessive, 2 little a's means they do have the disorder. Now, what we're looking at here is a pedigree. So let's talk about pedigrees and what pedigrees mean. Circle means female and a square means a male. The shaded in individuals have the disorder. This is an autosomal recessive pedigree. So we've got 3 individuals on the pedigree that are shaded in so that we know their genotype is little a, little a. We know that the father, in generation 1, the father, it has the dominant trait, but we are not sure if he is a carrier. So we don't know if he's big A, or big A, little a. So that we just put a blank there, or sometimes you'll see a question mark, but it'll just be big A, and then we don't know the second allele. We know he has one big A because he doesn't have the disorder. And that is, it's dominant to not have the disorder. Okay, so when we look down at generation 2, the original parents had 2 children, and they're both females. And then those females had children, and the first couple had a daughter, and the second couple had 2 sons and a daughter. Now, some of these individuals, we know their genotype, like big A, little a, or 2 big A's or whatever, but some of them we don't. And when we don't know the genotype, we just put a space there to represent that we don't know what that second letter is. But I also want to show you this.